Very good morning to all of you. So, total 19 only. Wonderful. 22. So, it is rising. The graph is rising. Okay. So, before we start the session, what would we talked about? That we have started with different type of mycoses. So, basically, different type of fungal infection. And if you remember, we have divided the mycoses or fungal reactions with respect to few aspects. With respect to few aspects means we talked about superficial mycosis, cutaneous mycosis, subcutaneous mycosis, right? And we started initially with a simple common example of dandruff. Okay. Now, as soon as we talked about the dandruff portion and what can be the different aspects of mycosis, we further move to an cutaneous mycosis, and under an cutaneous mycosis, we talked about an specific athlete foot syndrome, and then we talked about the tinea species. From tinea species, we talked about trichophyton and microsporum species. So we majorly covered how microbiologically we can identify trichophyton and microsporum species, and what are the culture characteristics of it, what are the mode of infections of cutaneous mycosis, tinea, trichophyton and microsporum. So majorly we try to analyze with respect to how does it infect, how we can identify it, what can be the probable mode of transmission. So we majorly had tried to find out all this uh, basic understanding on how the mycosis is spreading. Right now, once we are done with superficial cutaneous on the outer scalp and with respect to scalp, we have now entering into somewhat deeper aspect or the interior of our body. Once the fungal spores, fungal hyphae or mycelia enters into our body, the infection cause is much more severe compared to what we call as in superficial or cutaneous. And now that's why, because it is a severe infection, we call it as a systemic mycosis. Now, usually systemic mycosis affects the internal organs. It always affects the internal organs like lungs, kidneys. And the worst thing about the systemic mycosis is it spread in multiple sites of the body. Right now, usually if we think about bacterial yeast or any type of actinomycete infection, if it spreads, then also we have a specific antibacterial, anti-pathogenic, antimicrobials which can prevent, even though it is spread in the blood, it can dry the things, and then it is actually killing through a different mediation, right? Either cell wall, you talk about protein, or you talk about nucleic acids. Inhibition can be of many ways, but the problem with mycosis part is that because it is passing and it is connected to hyphae, it is connected to mycelium part. Due to this connection, actually, they are very, very hard to eradicate from the body, right? So that's why we cannot, once it enters into the body, we cannot omit the sporulating things, because if there are simple spores, then continuous higher concentration of any effective antimicrobial can be helpful to kill the spores. But how about our body has a multiple site for such spores? So what will happen at some point of time, that spore has a chance to propagate itself. Now that chance to propagate itself let's to and particularly higher side, that it can easily eradicate or it can easily spread into the body. And that's why the disease we are talking right now is called as an systemic, or it is called as a deep mycosis. Now how to eradicate it? Because it is connected through mycelium, it is connected to hyphae. We cannot directly just take out all the hyphae. And the much more problem comes is, if there is a chance that we can remove the organ, we think that there are much of the spores, we remove the organ. But how about sometimes it is involved, nervous system is involved into it. Now as such, because we cannot can cut neurals, or we cannot cut neural transmitters, so in that conditions this becomes very worse. It becomes just like an cancerous, where we don't know where the oncogenes expressions are going on. And if, because we don't know what the and how much extent it will is going to spread, we are not able to find out how much the systemic infection is going to be, that how much penetration power it would be with respect to its propagation, right? So that's why it's very essential that you go for a systemic mycosis and understanding the systemic mycosis better. Now, how does we differentiate this systemic mycosis? Because on the dermal part we have differentiated into a superficial mycosis, we have differentiated into a cutaneous, subcutaneous part. Similar to that, systemic mycosis is usually occur in three different forms, right? Now three different forms means one is called as an acute self-limited pulmonary. It is restricted up to some particular extent. Now it can be asymptomatic, we don't know it, that what can be the symptoms, and we don't take care of it, so it can be an asymptomatic, or else it from asymptomatic to very, very severe also. So from asymptomatic to severe conditions, it is always it is there. So that's why we found it that yes, from asymptomatic to severe conditions, it is something what we call as a self-limited pulmonary. Then there are some symptoms, and there is very high severity. Now due to very symptoms and high severity, it causes a disease which is pulmonary, or due to pulmonary, or it can be disseminated also. So it is actually a chronic, what we called it as. And then the third, what we call as a disseminated, that yes, it is there, it can be cured. Further, it can lead to a chronic, and mostly it disseminates in the body, right? So overall in dissemination, it is very, very hard that you identify any of the cases which are actually letting to dissemination process, or the fungal spores which are actually moving into a dissemination process, right? So that's why, first of all, what we need to understand with respect to systemic mycosis, the first thing what we need to understand with respect to systemic mycosis is we need to work with basic understanding of how does a systemic mycosis progress, right? So over there, first of all, we need to have an skin parasite. We need to understand that what can be the source of infection of systemic mycosis. For cutaneous or superficial, we understand that there are some skin parasites, and depending on the tone of the skin or the nature of the skin, it can infect us, right? But because we are talking about the systemic part, which is internal portion of the body, first of all, very essential is we need to identify the source of infection somehow. Now, once we identify the source of infection, the next thing that we need to understand is how does it is transmitted and what can be the susceptible host for that microbe would be, right? Now, how much it can infect and what can be the susceptible host would be. Anything or any human being or any host, any mammal can be a carrier, but the carrier, it is not just like a virus that carrier through a respiratory droplet can infect them or can transfer the mycosis. So the carrier for mycosis or systematic mycosis should be very infected with itself with a particular fungal infection. Otherwise, now it is not like that as soon as you sneeze it, you will liberate any type of spores. It is not like that. So for that, what we need to understand is what can be the probable source, who can be the probable or susceptible host. And yes, if we are able to understand all these things with respect to that, then automatically we can understand uh, what can be the reason behind the systemic mycosis. Now it has been found, it has been reported that these fungi who are actually an causative agent for systemic mycosis does not require any type of host like human being or animal to propagate itself. It can propagate itself within the soil. Now you can just imagine soil is full of different type of minerals. It is passing through many different type of factors. So as such, soil is somewhat having and many uh, cycles of infection. So if any organism is passing through that, right, if any organism is having susceptible or can survive in such infection or in such soil, now because everything is transmitted through particular portion, we are somehow going to be come into contact with the soil. We are tend, or human beings or animals are tend to have an infection of microbes. That is for sure, right? The only thing is now how much susceptibility of infection toward that host we don't know. So that's why for us to understand is the better thing would be that yes, fungi can exist, but it does not require our nutrient or our energy to survive. It can extract energy from everywhere, like a common example, soil, right? So that's why, first of all, how does we actually take up mycosis? That is most important. So either through inhalation, because we never stop inhalation, right? So we usually inhale the spores. Now, how does you think we are in? We cannot go to soil. I cannot go into farm and start inhaling, right? So, but what will happen through wind actually? The wind passes through different type of soil. Now this wind is coming into the atmosphere. Now within the atmosphere, we continuously, because we are inhaling and exhaling, we tend to take up the spores of this fungi, or else there is any cut in our body during like playing or during some particular or having some injury to our particular skin, it can be introduced to air also. So it is what we think the only inhalation. Apart from inhalation, this can be the also another particular thing where we need to see that yes, it can be there within the skin, right? And that's why first of all we need to understand what are the causative agents of such type of disease. Now, is there any specific causative agent for such type of mycosis? Yes, there are specific particular fungi, what we call as in blastomyces. Now, because it is blastomyces dermatitis, the particular disease caused by it is in blastomycosis. Conidioides immitis, the disease caused by it is called as an conidiomycosis. Histoplasma capsulatum, the disease caused by histoplasma capsulatum is histoplasmosis. And lastly, the Paracoccoides brasiliensis, and the disease caused by it is called as an Paracoccidioidomycosis. Right now, basically, the causative agent is this particular fungal species, Blastococci histo paracocci, and that's why mycosis is common. So osmosis, mycosis, mycosis, so suffix are common, and the initials of the suffix has been named, or has been named, sorry, named as per the causative agent for that specific disease, right? Now, what can be the particular this can include? This can include that there are various type of systematic mycosis, and that's why the respective etiologic agents, that's why whatever the agents which are responsible for the etiology should be considered separately despite of their similarities. So with respect to their similarities, we should always consider it is as particular different. Okay, so first of all what we will do is we'll cover individual particular mycosis part. So blastomycosis, then coccidiomycosis, histoplasma, paracocci. So starting with an blastomycosis part. Now blastomycosis is something associated with an endemic. Mostly it is restricted to in some particular region only, and that's why the name endemic has been given. It is something whenever any fungus resides on specific dead woods or dead things, it usually happens. And then the dimorphic fungus, which is actually been named, the fungus name is Blastomyces dermatitidis, right? Now, usually, if you remember, fungi or any type of fungi basically is usually cultivated at near about 25 degrees Celsius temperature, right? So at 25 degrees Celsius, usually we cultivate, and if you also incubate, you incubate at near about 25 degrees Celsius temperature, right? Now, blastomyces dermatitis is somewhat which is growing at 37 degrees Celsius. So talk, it is somewhere which is growing at 37 degrees Celsius, and it is something and around yeast like cells, right? So compared to a normal pure fungal strain, this is what we call as an pathogenic, because in order to get its pathogenicity, it needs to be survive at normal room temperature, and the optimal temperature where this organism can grow is room temperature, right? So that's why if we keep it at 37 degrees Celsius, we can easily get its organism to be cultivated. Now, how to identify it? Just like an ordinary microscopy we usually do, we go for its identification. We understand how and what the spore size would be, what the hyphae or form will be, and what can be the further. If we want to understand through a nucleic acid hybridization, we can easily identify it, right? So with respect to nucleic acid hybridization, we can find out what can be the cause of blastomycosis. So whatever the fungus can be, basically by microscopy or through DNA sequencing or through DNA hybridization, we can find actually what and how does a blastomyces dermatitis looks like. If we talk about its epidemiology, what is the particular distribution of it? Okay, Henson is asking, sir, I didn't get the reason blastomyces is capable of growing at 37 degrees. Yes, Henson. So why it has been marked at 37? Because for any of the pathogen, right? If we talked about the bacteria, we have gone through a different bacterial disease. Now, mostly bacteria is usually growing at 37 degrees Celsius. We also keep our cultures at 37 degrees Celsius, right? So normally, if we see normally our body temperature, and we talked about, is near about 35 to 37 degrees Celsius. Now for cutaneous and superficial mycosis, it is okay that the temperature is not mostly influenced. If we want to culture it, we usually culture it at 25 degrees Celsius or 22 degree, which where we get a maximum mature spores. Now, if we consider with respect to systematic, because it is within the body, any of the pathogen needs to adapt to that particular body temperature, right? So that's why this particular what we are talking about, the blastomyces species, is somewhat unique, because the optimal temperature of its growth is near about 37 degrees Celsius, and that's why it is much more chronic and infectious compared to other fungal disease, because it can easily adjust itself at that particular temperature, and it can spread within that particular temperature very well. So that's why this can be the one of the reason why it is growing at 37 degrees Celsius, and optimal temperature of that particular species at 37 degrees Celsius, right? So am I able to justify your question? Okay, okay, thank you, Vincent. So with respect to epidemiology, or with respect to what can be the probable geographic distribution of it, uh, the disease is somewhat what we call as an endemic, because it is not an epidemic or pandemic, but it is endemic. And where it is, you can just see it is mostly with respect to southeastern, like northern part of America, Ohio, like Mississippi, where much more frequency of infection with respect to blastomyces species has been noted down. So over there, the much more infections or might be infectious disease was causing with respect to its distribution. What can be the probable source of infection? Now, because if you see with respect to eastern part and southern central states, now if you just go through the American location and geography, much more disturbances with respect to environment in extreme conditions is happening on the eastern coast of America. Now, with respect to environmental disturbance means there are much more floods, occupational hazards, sometimes many particular torpedoes. So all major particular we talked about major disturbances with respect to plateau. If you understand the geography, plateau disturbances are most important. These are something majorly on the eastern part of the particularly America, right? So this that's why what can be the particular source of infection if we want to correlate it? The source of infection can be with respect to occupational or recreational activities which was found around streams or rivers, and due to this occupational hazard, too much of flooding, it causes a high amount of moisture condition. Now this high amount of moisture can definitely gives us a better soil fertility, but at the same time it also led to a better sporulation or sporulating environment for blastomyces species, right? So always think that wherever there is a high amount of moisture will be there, the chances are that this particular species will grow much more and the sources of infection would be much more, right? Now, what can be the pathogenesis and clinical picture behind it? The clinical picture and pathogenesis would be then blast mycosis. Now, what can be the blast mycosis? Usually through inhalation. And if you understand, there is an dimorphic characteristics of one of the organism we talked about. Now, what can be the dimorphic characteristics is sometimes hyphae is converted into an yeast form by just a temperature difference, right? So similarly over here is that whenever we are inhaling the sporulating form, once it is inhaled and it is entering into a body, the dimorphic form of that particular fungus comes out. So the dimorphic form would be it is converted from the hyphal part to an yeast part. So from hyphae to yeast, it once it enters into the it converts into that particular form. Now, once it is taking some time for conversion from hyphal to particular yeast form, normally it takes 30 to 45 days for an starting of acute pulmonary disease, right? So normally acute pulmonary disease is taking near about 30 to 45 days, and usually it is very indistinguishable. We cannot easily distinguish it between an bacterial pneumonia and particularly fungal pneumonia part, right? So that's why we cannot just easily distribute or we cannot easily understand it. However, the good thing or advantage of this particular process is at least, let us consider at least 50% are asymptomatic disease. We don't understand the symptoms. It eradicates from the body itself. Right? It is something asymptomatic. We never take care of it, and it eradicates itself within the particular body. So 80% or 50% is really asymptomatic. However, rest of the 50% which are infected, right? It goes into a chronic form. Now, chronic form means it can affect various multiple organ site in our body. Now, multiple organ site means it is going to infect lungs, skin, genitourinary tract, reticuloendothelial system. So, with respect to every particular such type of particular disease or manifestation, it is going to like very severe and chronic, right? And what can be this? This is what we call as in blastomyces. It looks like, you can see the particular portion of the finger. It is totally been covered with a black spot. So now just think about such type of incidents, and if this happens, how can we eradicate it? Because there is no specific cause for it, and there is no specific this specific medicine for it. Now why I'm talking, because if I just apply any of the particularly antifungal agent, it cannot kill. It kill up to this much extent of spores, let us say. But how about the small spores which are germinating at that time, right? So because it has spreaded, we cannot know it. So usually the infection with respect to cutaneous, like uh, it can happen with in combination of other disease also. uh, Now what can be the combination of other disease means, sometimes pneumonia, which is we all know because it is in either step, pneumonia has been infecting. Similar type of symptoms whenever the chronic blastosis happens or blastomycosis happens. Similar symptoms like fever, cough, myalgia, myelitis. But because it is something which is in correlation with another type of species, usually it has been found that it can be treated and it is usually been cured also. It has been found that it can be cured easily. However, whenever it goes into a severity stage, now severity stage, the infection actually appears in three different forms. Now, three different forms means it goes into an acute pulmonary, it goes to a chronic pulmonary. Now, what is acute means? Even though it may be resolved without treatment, however, the treatment is often been prescribed by the doctor. It is something like an asymptomatic. Usually, person carries the mycosis infection, but it is asymptomatic, and due to such asymptomatic infection, person throughout the lifetime does not recognize about it, right? If we talked about the chronic part, it is something what we call as a granulomatous lesions in upper lobe, and it can be sometimes misdiagnosed as a cancer, right? Because it is generally similar, giving similar symptoms associated with it. That's why it has been shown over here, right? So if we are talking about such chronic part, it is first of all biopsy is required. After biopsies, you need to go for an culture report, and through culture report only we are able to identify, yes, it is and mycosis, not actually what we call as an carcinoma or cancer. The third form of the infection is called as an disseminated, is something which is a progressive disease, and it follows something unresolving pulmonary lesions. We are not able to, through even biopsies, we are not able to identify that, yes, this is what actually in pulmonary disease are. So we will be not able to identify what is the cause and what is actually happening inside our body, right? So that thing we will never able to understand what and how it is happening. Plus, mostly you will never heard about it. It affects our bones, skin. Like skin can be cured through a dermal particular antifungal ointments or antifungal creams. But how about the bone density has been changed, right? That the bones are most affected with such type of disease. So uh, bones becomes hollow sometime, it becomes brittle. This is what we call as a disseminated blastomycosis, that through an progressive infection, we ultimately don't know what would be the end of that. It leads to a brittleness of bone, and ultimately bones are deteriorating. And once the bones are deteriorating, we cannot help ourselves, right? So how do we go for a laboratory diagnosis? Just like a normal what we have discussed in our cutaneous part and superficial mycosis or dermatitis part. Same thing, either we can go for an KOH, that we prepare a 10% KOH, put the fluid into it and examine that yes, the spores are there and what type of sporulating forms are there. Usually, if we see under a microscope, the fungus occurs like an thick-walled globule yeast with 8 to 15 micrometer in diameter. So micrometer diameter is usually somewhat like that. And each blastoconidium is attached to parent cell to a broad base, right? For even though it is somewhat 8 to 15 micrometer, sometimes it is been confused because of similar structure resemblance with an Candida imitis and Candida neoformans and Cryptococcus neoformans. It is really hard to identify through microscope, and at that particular time, usually it has been identified through nucleic acid hybridization or else DNA sequencing part. Okay. Can you please repeat on examination? Examination part. Okay, yes. So just usually what we used to do in superficial or cutaneous mycosis, we always examine basically culture using a 10% KOH solution, right? It fixes the particular spore, so the person who are actually observing such spores does not get infected. So potassium hydroxide is something what we call as an alkaline, and usually the fungi is growing at acidic pH. So as soon as we are changing the pH and environment, it can be easily observed. But the problem is because of its dimorphic character, it resembles the characteristics of yeast. Right? It is exactly resembling the characteristics of yeast. And due to such resemblance, uh, how does we observe it? First of all, we need to observe microscope. Like usually, if we see the diameter and measure the diameter, it is somewhat 8 to 15 micrometer in diameter. Each particular blastoconidium is attached to an parent cell at the bottom, right? But the problem with this particular identification is because it is resembling similar to yeast. There are yeast which we call as an seed Cryptococcus neoformans, or there is also an Candida. So these two Cryptococcus and Candida is similarly resembling 8 to 15 micrometer with respect to blastomyces. And that's why it is very hard to distinguish between both the cultures of blastomyces dermatitis and Cryptococcus or Candida imititis, right? So that's why it is usually been, if we want to differentiate it, the best method to differentiate it using a nucleic acid hybridization. And if not nucleic acid hybridization, we can go for DNA sequencing. So specific 5.8 ITS intertransgenic spacer arms, if we are able to identify or conserve region of it, amplify it, then we can easily identify that particular culture. How does, if we want to go for in which media we should use, we should usually use Sebrod glucose agar or Brait Hart infusion agar, yeast extract along with an cyclohexidine, then it will be incubated, and then the culture should be kept for at least 28 days uh, before we found it, yes, it is a negative or positive, right? So what can be the different two phase? If we observe it, there are two phases because of its dimorphic characters. Now, what are that two phases? The two phases will be what we call as in mycelia phase and yeast phase. So on sabrodextrose or sabroglucose agar, if we observe the colony, it looks like white, similar to yeast, but it then turns into cottony because of aerial mycelia, and further it becomes, as it becomes mature, the pigmentation, brown pigmentation is observed in sabrodextrose agar or sabroglucose agar. If it is in yeast phase, right, if the organism is in yeast phase, then in yeast phase, we need to cultivate in brain heart infusion agar, and as soon as we cultivate in brain heart infusion agar, the colony appearance is waxy, tan, wrinkled, and what we need to do is we need to inhibit the organisms, or what we call as an bacteria, using an clonophenicol or cycloheximidine, right? So this is how actually it looks like whenever you go for enzyme sustaining. This is what we call as an budding yeast, and in mold phase, similar type of structure has been seen, but it is based is something what we call as an now, this mycelia is the base, so what we call as an broad base, and over here it is like this, right? So hopefully we stop over here. We'll continue with blastomyces dermatitis in our next session still. So those who are interested can search. It is very, very nice that one organism can have a different dimorphic character whenever it enters into host, right? So what it gives is how an organism can adapt to the environment, even change its shape, even degrade the mycelia and convert it into an yeast-like form. It is a very, very interesting subject to read, that how organism can phenotype change or phenotypic characterization or plasticity is there. Just understand this is what we call as an plastic behavior. So there is an plasticity observed, and due to this plasticity, it has been found that this is important with respect to pathogenicity, right? So before we stop our session, I again remind you it is compulsory for all of you to attend the orientation session. It won't be more than half an hour or 45 minutes. So I had shared the link. Your placement orientation will start at 12:30, right? So it is online, so it is mandatory to attend it. It will not be more than 45 minutes. Please attend it, right? Okay. With respect to that, I am speaking of the name who are attending the session. Uh, just a second. Okay, so starting with Anusha, Brijesh, Daksh, Devanshi, Dhruvi Chandarani, Dhruvil Chaniara, Dikshit, Diya, Fahad Gada, Hanson, Hasti Thumar, Hetal, Hetvi, Himanshu, Ishika, Jaisalba, Kennedy, Khushi Parmar, Khushi Parekh, Krishna Maradia, Krishna Vania, Mandeep, Mansi, Mark, Masti, Mitali, Mutebi, Nancy, Nandini, Narendra, Nehal, Nelson, Nikita, Om, Paras, Parthrachi, Priya, Priyanka, Raj, Rajvi, Nitin, Riya, Ronnie, Roshni, Shakir, Saurav, Shraddha, Shruti, Shubham, Sun, Tanya, Tejas, Tito, Umul, Vivek, Vivian, Wilson, Esha, Zara, Ritika, and Vijay. Okay. So with that, thank you all for attending the session. Thank you. We'll see you on Monday. Thank you.